All right, hello biology people, here to continue with our chapter 6 discussion. I hope y'all are doing well. Hope you've watched the other prior videos talking about fatty acids, talking about lipids, different lipid categories. So now we move into an important part of chapter 6, which deals with membranes. Right? So if you recall, our fatty acids, which can be of one or two categories, saturated fatty acids or unsaturated fatty acids. So that discussion is very relevant for today as well. Instead of looking at 3 fatty acid chains, now we'll be only looking at 2 fatty acid chains. And we bring back a functional group from chapter 2. If you recall our different functional groups, the aminos, the carboxyls. Now we look at a phosphate, right? So phosphate functional group. Some other molecules here, but at the bottom we then have 2 fatty acid chains. Right? So this is in the middle, a little bit more realistic diagram of a phospholipid. Some molecules. There's a phosphate, the glycerol, which we've talked about before. We saw glycerol with the triglycerides. So now we have the glycerol connected not to 3 fatty acids, but only 2. And again, through the process of dehydration synthesis, we connect these 2 together. In this situation, if you remember that linear molecule, that becomes then our saturated fatty acid. We have a kinked or unsaturated fatty acid. This is just an example. They can be both saturated, they can be both unsaturated, or one of each. So just the way that artists drew it there. So this is like a computerized version of the molecule. Here's a little bit more simplified view. Little zigzag, yellow rectangular fatty acid representing a saturated. Here we have an unsaturated. And if we want to get really simplified, really lazy, we can show it with a little circle representing the phosphate area, and then these 2 little sticks representing the fatty acid tails. Now, the book gets into some vocabulary here. They're going to mention the term hydrophobic and hydrophilic. Right? So hydrophobic and hydrophilic. If you know of a phobia, right? A phobia is a fear, a very strong dislike towards something. Well, the fatty acids, because they don't mix with water, they're claimed to be hydrophobic. They have a phobia of water. They separate on top of water. So these fatty acids down here would be considered nonpolar or hydrophobic. So hydrophobic tails. And the top part, if you notice up here, there's a positive charge, there's a negative charge. So there is some electrical charges that would be attracted to the delta charges of water. So water would interact very easily with the phosphate area. We're going to call that then a hydrophilic area. So hydrophilicity and hydrophobicity here. So the hydrophilic area and the hydrophobic area. Hydrophilic head, hydrophobic tails. Hydrophobic tails and hydrophilic head. Hydrophilic head, hydrophobic tails. So I want you to practice that vocabulary. You will see that vocabulary on the next exam. Just make sure you understand where that pertains to the physical location of these characteristics as well. So if we were to take a bunch of these phospholipids and just throw them in water, right? Does it make sense that they would orient themselves like this top picture? So here we have water, we have the hydrophilic heads in relation, in communication, in contact with the water, and then the hydrophobic tails floating on top, not actually touching the water, right? So I hope that orientation makes sense to you because of the hydrophilic or polarity, or hydrophobic or non-polarity areas. So again, this would happen in an area like a pail of water, something like that. But what if we go inside of the body? There's water in many parts of the body. So we know we have plasma, so there's water in the blood. We haven't covered this yet, but we're going to have water inside cells we call cytoplasm. We have lymph. We have urine. There's all kinds of water in the body. So if you are a phospholipid that finds yourself in the body, surrounded by water on all sides, they start to orient themselves something like this. So there's water here and water here. This is the most comfortable position, most comfortable orientation that these hydrophilic, hydrophobic regions can attain. So again, the hydrophilic heads in contact with water, hydrophobic tails kind of hidden away from the water by the head region there. Right? So again, we are not going to call this a single layer of phospholipids. We use the term a phospholipid bilayer. So 2 layers. That the top layer, not in direct contact. You notice that space. So this layer is not in contact with that upper layer. So the layers can move left and right independently of each other, forward and back independently of each other, sideways. So again, 2 independent layers, but making up 1 phospholipid bilayer. And again, more realistic sort of detail here. So we would have the hydrophilic head, hydrophobic tail, hydrophilic head. And we would have, again, these 2 layers. And again, these are going to start to comprise biological membranes. So membranes are made up of this phospholipid bilayer. And the whole point of these is to regulate what crosses from one side to the other. And that's going to be an important role. So also embedded within these phospholipid bilayers are these sort of tubular type of proteins. We talk about proteins, we talk about the alpha helix. Well, we have proteins with very, very well-defined alpha helices that allow the entire width of that protein to span the entire width of that membrane. We're going to call these integral membrane proteins, or maybe IMPs. Integral membrane proteins will traverse perpendicular here, or I should say they're parallel to the fatty acid tails. So they're the whole length or width of that membrane there. So IMPs traverse the bilayer, and they're going to be scattered throughout the entire sort of membrane. So there's our tertiary protein, has to be functional, but made up of many of these alpha helices. And in essence, they form little tunnels, right? They're going to make little tunnels that then allow material from the top part of the membrane to kind of move through easily to the bottom part of the membrane. So the whole point of their existence is to allow a passageway for molecules to move through. And they're going to facilitate then the movement from one side to the other. It would be very difficult for things to pass. It is possible, but difficult for things to pass through this part. Right? If you kind of visualize the chemical qualities that something would have to have. So it would have to be both polar to interact here, and then it would have to be non-polar to sneak through here, non-polar, and then polar. And again, it's difficult for big molecules to have both of those qualities. Maybe if they're very tiny, they can sneak through, kind of squeeze through, but it's going to really be difficult for larger particles to move through that membrane. So larger particles will have to pass through the integral membrane proteins to cross that membrane. So we're in a sort of a geographic location here that we're sort of a sister city, if you will, to Juarez, El Paso and Juarez. We're very, adjacent to the border here. So if we analyze these bridges, right, these bridges are going to serve the same purpose as do these IMPs. They're not tunnels, but they allow for sort of guided movement from one side of the border to the other. And by channeling all of the traffic through here, it allows for a much greater checking, regulatory sort of aspects. We can kind of analyze each individual car that passes across, and that's going to be an important characteristic of this bridge, an important characteristic of this sort of little tunnel, this integral membrane protein. So it allows for regulation. If things are sneaking through over here on the sides, we may not know what's passing from one side to the other. So again, a couple of other little puzzle pieces here. So when we look at this entire phospholipid bilayer, you're going to start to hear another vocabulary term. They're going to call the plasma membrane. So the plasma membrane is what surrounds in 3 dimensions all of the cells. So plasma, because they're not quite solid, they're shifting, they're fluid, but it's not quite liquidy. So it's got that sort of intermediate type of texture to it. They're going to help regulate movement in and out of the cell. They regulate what goes across that membrane. And again, different artist rendition, different colors, but we have our blue area, the hydrophilic area, the little black tails, the hydrophobic area, and then again, the blue hydrophilic area. We also have these big purple blobs that represent our IMPs, so integral membrane proteins that allow for movement. Now, if you're retaining all the information that we've covered, if you see these little things here, these little hexagonal shapes, I hope you remember what those little hexagonal forms are. Those are carbohydrates, right? Carbs. They're between 3 and 20 monosaccharides joined together, and they're marking the surface of these proteins or the surface of the cell. So what are these little carbs? These are the oligosaccharides. And again, I hope you remember those. Oligosaccharides not used for energy, but for cellular marking. And they're going to be on the outer surface of the cell. So that tells us this is the extracellular area outside. This would be the inside of that cell. So let me back up to the very first slide. This gives us a realistic view of what this membrane would look like. Right? So here we're in the inside of the cell. If you're in the inside looking up, just like if you're inside your room, you look up, you see your roof, right? Or I should not say the roof, the ceiling. So you see the ceiling in that roof, and the ceiling in that room. So we have all of these little hydrophilic head regions that you're seeing. We don't see the tails, we don't see that other layer. So if you're in the inside, all you see is the hydrophilic area. If you're on the outside, all you see is that top hydrophilic area. We will see also some IMPs that are going the entire width of that membrane. And then on the outer side, or the outside outer surface, we have our little oligosaccharide markers either attached to the proteins or attached to the hydrophilic head areas. This is for another discussion later, but we're going to start to see how these IMPs can actually start to connect to other cells to allow for multicellularity. So that will, again, a future discussion. But for now, focusing on the actual phospholipid bilayer. So let me pause this one here, and then let me continue with another discussion on diffusion.